Welcome to CBSE NCERT Solutions by Vedantu. I hope I'm audible, visible, and the presentation also is visible. Please let me know in the chats if I am audible and visible, and even I'll be checking the chats as well. So welcome all of you to Molecular Basis of Inheritance Part 3. So we have completed Part 1, we have completed Part 2, and those students who did not find these parts, go to the video session and check it because it was recorded, okay? And today we are going to do the third part of molecular basis of inheritance. It's actually a very big chapter, so usually it takes up to maybe 6 to 7 hours to go thoroughly with the chapter and also learn go through every lines of NCERT, okay? But I have done it in 3 parts, okay? I've done it in 3 parts. So let me check. I think ad is playing, so we are live. Voices also chapter and also learn go through every lines of NCERT. Okay, fine. We are audible and visible. Everything is perfectly fine. Let me just connect. Okay. Fine. So molecular basis of inheritance part 3. Okay, let's start with part 3. So yesterday's class we had done up to transcription. Okay, transcription. What was transcription? Very simple. When DNA becomes mRNA, that process is going to be called as transcription. That process is going to be called as transcription. Yes or no? Now mRNA to protein, it is going to be called as translation. So this process of DNA becoming mRNA is called transcription, and that is what we have learned yesterday's class, that transcription, how does it happen. Now today we are going to learn some other topics. Before we learn translation, you have to understand that, hi Tanuja, you have to understand that there are genetic codes present. Okay, so we are going to learn how this translation happens, or what is how is the reading going to happen, how are they going to form protein, how do they know that they have to code for this protein. All that we are going to learn. Okay, now see, in eukaryotes there are 2 additional complexities. That is, there are at least 3 RNA polymerase in the nucleus in addition to RNA polymerase found in organelles. There is a clear-cut division of labor. Now clear-cut division of labor, what they're talking is, students, to remember this, there is a trick called RMT. Like how you remember your MTR and all of that. No, remember RMT. RMT means, and then put 1 because this is in the first position, then put 2, then put 3. Now ma'am, what have you written? So look here, RNA polymerase 1, okay, all these are RNA polymerase numbers. RNA polymerase 1 codes for rRNA, okay. RNA polymerase 2 codes for mRNA, mainly, okay. There are other subunits that they will code for, but mainly they'll code for mRNA, and RNA polymerase 3 will code for tRNA. So if you see anywhere RNA, mRNA, tRNA, and they have given a match the following asking you which type of polymerase it is, you have to be sure that this is the trick to remember, RMT, right? RMT, then write 1, 2, 3, then you will be able to remember that, okay, polymerase 1 rRNA, polymerase 2 mRNA, polymerase 3 tRNA. Okay, so this is what they are going to tell you. See, look here, RNA polymerase 1 transcribes rRNAs, okay. That is, see, 20S, 18S, 5.8S, whereas RNA polymerase 3 is responsible for the transcription of tRNA, 5S RNA, and snRNA, which is a small nuclear RNA. So students, this is a very, very, very important PYQ for my NEET students, and for both students also this can come as a PYQ, but for NEET students this is mainly a PYQ, okay, where you have tRNA, sRNA, and snRNA and 5S RNA, okay, 5S RNA. So now the RNA polymerase 2 transcribes to the precursors of mRNA, the heterogeneous nuclear RNA. Okay, heterogeneous nuclear RNA, that is hnRNA. Okay, this I told you yesterday in class, and I have mentioned that exons and introns will be there. So it is going to be a mix, right? It's not a homogeneous mixture, it's a heterogeneous mixture. So it is called as heterogeneous nuclear RNA. Now the second complexity is that primarily transcripts contain both the exons and the introns and are non-functional. Hence it is subjected to the process called as splicing. Okay, it is subjected to the process called as splicing, where the introns are removed and the exons are joined in a defined order. So I told you this yesterday's class, no, that what is going to happen if this is the, okay, if this is the, I think this color is visible. Yes, look here students, is this two colors very differentiable or should I use another color? Okay, this, I think I'll use another color because it's not that differentiable. Let's go for blue. This is total differentiable. See, these are the two colors, right? So now I'm not doing a coloring thing, but understand these are the exons, this is the introns, again exons, okay, introns. Got it. Now what is going to happen, this introns are actually something that I don't require. Okay, now if I don't require them, I'm going to do a process called as splicing. Okay, splicing, where I am going to remove these, okay, I'm going to remove these introns outside and I'm going to form a strand that is only with exons. Yesterday I had told you this. Okay, I'm going to form a strand only with exons, an mRNA with only exons. Now this mRNA which has only exons, okay, the mRNA which has only exons are going to be the ones who are going to code for a protein, because these introns are non-coding sequence. So when I am going to code, when I'm going to make the protein, do I want the coding or the non-coding? I want the coding sequence. So exons are the coding sequence, introns are the non-coding sequence. So we do not want the non-coding sequence. So what we are going to do, with the help of splicing, we will remove those people telling that you are nonsense, we do not need you, and we will make a mRNA which has only the coding sequence. Okay, now students, since the coding sequence is there, we also see there are multiple reasons. Okay, so I have read a lot about this. So there are multiple reasons for why capping and tailing is done. Okay, so some times it is told that capping and tailing is done so that there is no add-on of anything, like no add-on of introns, no add-on of any other, you know, undesired things. Okay, then I have seen in one place they tell that it is to basically label the 5' end and 3' end. Okay, such things keep happening because in DNA we know that there is a, on the 5' end there is a free phosphate group, in the 3' end we have a free OH group. Now in mRNA we have to see that. So to make that differentiation between 5' and 3', we have to add something, no? So like that. See, hnRNA undergoes additional processing called capping and tailing. In capping, an unusual nucleotide called as methyl guanosine triphosphate is added to the 5' end of the mRNA. See, 5' end is already known. So statements like this telling why we do capping and tailing, you know, it's kind of little confusing. In the tailing, we have adenylate residue, that is 200 to 300 are added at the 3' end in the template independent manner. It is fully processed hnRNA. So to even do the entire processing also, we can do this capping and tailing. So we feel like, okay, this is a complete RNA, like complete RNA. There is no, like, what to tell, there's no, like, it's not incomplete, only the sequence is made, the capping and tailing is not done. So that kind of different you know, answers will be there. And also they tell that it is this capping and tailing is done so that it completes, and then this will be transported to the cytoplasm, because it now the mRNA that is transported out of the nucleus for translation. So students, if the location is asked to you, always remember that your transcription, replication, that is your in your central dogma, your replication actually you can leave. Okay, transcription mainly, students, transcription happens in the nucleus. Okay, transcription happens in the nucleus. So let's say if this is the nucleus and this is the cell, this is the cell. Okay, oh, it went outside. Let me draw it here, small cell. Okay, I'm drawing it here. Okay, let's say this is the cell. Okay, and this is the nucleus. inside this nucleus that mRNA formation is going to happen, but after it is formed, it is going to be transported. Let's say this is the mRNA. It is going to be transported outside and it is going to come to the cytoplasm. So here is where your translation will happen. Okay, in the cytoplasm is where your translation will happen. So this you have to remember and keep, because location might be asked, and when location is asked, you shouldn't be like, oh, where did it happen? Because understand, double-stranded DNA, okay, we form a single-stranded RNA. This transcription is going to happen inside the nucleus itself. Okay, transcription is going to happen inside the nucleus, but then translation is going to happen here. Okay, translation is going to happen here. I hope all of you are clear and did not see the chats. Chalo. Okay, it's clear. Fine. Okay, likes are not increasing. Why so? Let me tell them. Such confused people. Okay, told. Okay, I think so many ads are playing and students are getting disturbed. Okay, but I don't know why is it that nowadays ads have become more. I think it's also to make everyone convert to premium, because there's a lot of YouTube users, no? So yes, students, look here. If determining the biochemical nature of genetic material and the structure of DNA was very exciting, the proposition and the adhering of the genetic code were almost most challenging. Now, before, NCERT has a problem. NCERT has a problem is that NCERT does not think that you do not know anything. NCERT thinks that you have already studied and you're chuma doing time pass, like reading NCERT. So sometimes connection goes, okay, but it's an amazing set of book which knows that, okay, what should be the content, what should not be the content, but there are some issues like this. So let me tell you that genetic code, before I introduce you to genetic code, understand that your genetic code is something that will be on your mRNA. So now all this while we were talking about mRNA, but we never spoke about what is in the mRNA. So students, mRNA is going to have something called as genetic code, which are triplets. Okay, so we are going to learn how they became triplets and why it is triplet, not something else. Okay. So here I have written some, codes. Now students, these codes are present on the mRNA. These codes are present on the mRNA. Now, if these codes are present on the mRNA, someone has to read it and tell that, okay, AUG is a start codon, or AUG is the one who is going to code for the protein methionine. Okay, it is going to code for the protein methionine. Now, who is going to tell that? For that, we need some set of people. So, we are going to learn that. Okay, we are going to learn that. So, students, see, even though transcription, translation, everything, finding was a smooth process, they found a lot of difficulty when it came to finding the genetic code. So in a very true sense, it is required involvement of the scientist from every discipline, that is your physicist, organic chemist, biochemist, genetics. It was George Gamow, a physicist, who argued that since there are 4 bases, if they only code for 20 amino acids, the code should constitute a combination of bases. Okay, a combination of bases. He suggested that in order to code for 20 amino acids, the code should be made up of 3 nucleotides. But this did not come like this. Okay, randomly it did not start like this. So how it started with students? So a person, a physicist, okay, who and all is coming in biology, you see, a physicist called George Gamow. What did he tell? He was like, see, we have 4 bases. Yes or no? We have A, G, U, C. These are the 4 codons or 4 nucleotides that we have. Now, with this, we have to make a combination that will code for 20 amino acids. 20 amino acids. Now, first, many people told, let's do one thing, 4:1. That means each of them will code for 1. Obviously, we are going to get only 4 amino acids are going to get coded. Then they told, let's do one thing, let's do 4 2s. Okay, that is 2 combination like AG, UC or GU. Okay, like that. Okay, AC. Like that, let's code for, let's make 2 combinations. So 4:2, this is permutation and commutation. Okay, maths also I'll teach. So it is going to code for 16 amino acids. Okay, A should always be small. Okay, 16 amino acids. But how much amino acids I have? 20 amino acids I have. Now I have coded for only 16. That means 4 is left. So can I leave the 4? I cannot leave the 4. Then George Gamow told that let's do one thing, let's code for 4:3. That is 4 into 4 into 4, that is going to be 64 codons. How much I have actually? I have 20 codons, but I got right now 64 codes. They were like, it's okay, more the better, more the better, because students, if we go vice versa, or if we go with these two positions, then you should understand that these two are less. These two are not even coding for the ones that we require, that is 20 amino acids also, it is not coding, so it is going to be less. So instead of that, why don't we do something in the position that let it be extra, no problem, let it be extra. So this is how 64 codes came. Now in the 64 codons, students, what is codon? I just told you, no, it is present on the mRNA. Okay, it's present on the mRNA, and whatever is present on the mRNA, you have to remember that these, see, this out of 64 codons, 61 of them, okay, 61 of them is going to code, okay, code for amino acids, but 3 of them are actually nonsense codon. Nonsense codon. Why am I telling nonsense codon? Because they are not going to code for any amino acids. They are actually stop codons. They are actually stop codons. Stop codons means who is going to tell that, okay, enough, this is time for you to stop this reaction, okay, just stop this. So say out of the 64 codons, 61 codons are going to code for amino acids, and 3 of them are nonsense codons who is going to stop or tell that, okay, ruk jao, okay, like enough for today, like that, okay. So this is how codons are there. Now students, codons, try to understand, they are a combination of 3 amino acids, because we understood, no, 4�3 means what? I have to make a combination of like AUG, okay, I have to make a combination of UAG, like it will always be 3, and this one combination of a triplet will code for a protein. How it will code for a protein? First it will code for an amino acid. Many amino acids combine to form a polypeptide chain. Okay, polypeptide chain. Yes or no? Got it. Okay, chalo. So this kept changing. Okay, this kept changing. Why so, Jayanti? Jayanti is telling me better. Okay, now providing proof that codon was a triplet was more daunting task. The chemical method developed by Hargobind Khurana was instrumental in synthesizing RNA molecules with defined combination of bases, that is homopolymers and copolymers. Homopolymers are nothing, students, like this, okay, like this. Homopolymers are like this, A. Okay, people will think I'm crying or something. U. G. That means what? They are homopolymers. Okay, homopolymers. Got it? They could be homopolymers. Okay, then copolymers means what? Copolymers means AU. It's like hey you. Okay, and then GC, something like this. Combinations I'm telling you. GC. These are copolymers. So they first started with this, then later they found out, okay, this is not, we will do a combination of 3 and all of that. So this Hargobind Khurana was instrumental in synthesizing RNA molecule with defined combination of bases, where he told homopolymer and copolymer. Okay, then we have Nirenberg's cell free system for protein synthesis finally helped in the code to be deciphered. Okay, Sevidio-Oho enzyme, that is polynucleotide it's not coming in flow. Polynucleotide phosphorylase was also helpful in polymerizing RNA with defined sequence in a template independent manner. Okay, enzymatic synthesis for RNA. Finally, a checkerboard for genetic code is prepared. So students, here what you have to remember is, you remember this, what did Hargobind do? He did homopolymers and copolymers. Nirenberg came up with a cell-free protein synthesis, telling that, okay, let's do one thing, let's make codons. Okay, let's make codons where you do not need the whole cell, and we'll do a cell-free system for protein synthesis. Finally helped in codon. Okay, and then an enzyme was formed, which is a polynucleotide phosphorylase, which was helpful for polymerizing the RNA molecule. You do not have to know the details, just remember that you have to just know this 3 scientists name. Anywhere you see the scientist name, you should be in a position to answer. Now, ma'am, what is genetic code? How did they form? So look here, students. See, this is like your how your Punnett square is there, no, in your principles of inheritance, Mendel's, like that. We put all the 4 codons in all the 3 positions. Okay, this is a different thing for you guys because you always do 2 positions, right? This is how your Punnett square is going to be. Okay, this is going to, it's now say AA. This is how it's going to be, right? 1 position and 2nd position. There's no 3rd position. So here there is going to be a 3rd position, and look at the 3rd position. In one box only all the 4 is going to come. Next box all the 4. Next box all the 4. Next box all the 4. It's not like this. It's not like this. Here only you will get U and G together. Okay, if I put U and G, you might get confused. Here only you will get A and capital A. Here only you will get capital A and small a. Understood? Like this the boxes are. So 3 positions are there. Clear? Chalo. Now students, look, you do not have to learn this box luckily, but you have to know maybe some very common ones. Okay, very common ones. So look here, U, 1st position, 2nd position, 3rd position will land you in this. Same way, 1st position, 2nd position, this one will land you in this. Same way, 1, 2. So let's label this 1, 2, 3, 4. And let's label this first 2. Okay, 1, 2. So it is going to be 1, 1, 1, 1, 1, 2, 1, 1, 3, 1, 1, 4. This makes the first box. Got it? So you do not have to know the all of this, but just remember that you have to know how this formation happened. Okay, so here students, what you have to focus on this UAA, UAG and your UGA. These 3 are the stop codons. These 3 are the stop codons. Stop codons means what? Who is going to stop the translation from happening? So here UAA, okay, UAG, UGA. Okay, this is the combination. These 3 are the nonsense codons or the stop codons. Are you all following? Yes or no? Hi Nishant. Now, since you don't have to learn the codons, you have to learn something else. You have to learn the salient features of the genetic code, or how are these people formed, other things you have to learn. Okay. Now see, the codon is a triplet. Yes or no? That is formed of three units are there. Out of that, 61 codons code for amino acid and 3 codons do not code for any amino acid. Hence, it is a stop codon. Some amino acids are coded by more than 1 codon. Hence, code is degenerate. That is, some amino acid students are coded for more than 1 codon. That is, like let's say, let's just go here and see. See here, look here. Phenylalanine is going to code for this. Okay, somewhere else also it's there. See, two codons. Understood? See here also, histamine. Here also, serine. Like that, valine. Like that, see, valine. So that means one amino acid can code with two different codons, okay, 2 or 3 different codons. So some amino acids are coded by more than 1 codon, hence the code is degenerate, okay, hence the code is degenerate. The code or the codon is read in mRNA in a contagious fashion. The codon is read in a contiguous fashion, okay. I just finished human health and diseases, so that words are coming for me. Let's say AUG, UUU, UGA. Okay, AUG, now I've written in 2 pattern. Yes or no? I've written in 2 pattern. Look at this pattern. AUG, comma, UUU, comma, UGA. No, wrong. You read it continuously. That is AUG, UUU, UGA. So if these codons are present, no, they are going to be read like that only. AUG, UUU, UGA. If one fellow goes also, the next fellow will start reading it. Okay, it is not like, okay, now imagine this G went off. A will come here, next person will keep continuing the reading. It is not like it is going to stop. Okay, it's not like it's going to stop. Now look here. The codon is read in understood no continuous manner. There is no punctuations. The code is nearly universal. For example, from bacteria to human students, you will code for phenylalanine only. It is not going to change. Be it you are the smallest organism, be it you are the most complex organism, what you is going to code for phenylalanine only, no matter it is from bacteria to human. So it is universal for every organism. From ant to elephant, same thing is going to happen. Understood? So some exceptions of this rule have been found in mitochondrial codons in some protozoans. Okay, only in some protozoans we have found some exceptions. Now AUG has dual functions. So some of the codons, especially your AUG, have dual functions where it is a start codon. At the same time, it's also going to code for methionine. It's also going to code for methionine, that is MET. Okay, methionine. So AUG, if we tell, it will also help as an initiator codon that is going to start the reaction. At the same time, it's going to code for methionine. Understood? It's also going to code for methionine. Now UAA, UAG and UGA are stop terminators. Okay, stop terminators. So remember like this: UAA. Okay, UA, Yuga, Yuga. Okay, Yuag, Yuga. Okay, now there's a very classic example of your point mutation. Now before I get into mutation, try to understand that even, let's say, okay, let's just see the combination. Okay, GAG went up to coding for it just I've written just two things. Okay, imagine this was the one that was coding for glutamate. Okay, this was the one that codes for valine. Now students, you will be like, ma'am, just one letter change, what's the big deal? Right, one letter change. This one letter changed. Adenine got replaced by uracil. This one letter change, I have written it as a letter, but imagine in your body it's going to be a change in the base, okay, or change in the nucleotide, right? If students, change in even one nucleotide can lead to mutation. Now the example that I've written is the example of sickle cell anemia. Sickle cell anemia, that is a very dangerous thing, and it can come in for like one, it comes for one person among thousands, 10,000 people. So it's not a very common disease as well. But sickle cell anemia, you understand, where glutamate should have coded, their valine got coded, and it led to point mutation. So mutation can be point mutation where one nucleotide change. Okay, one nucleotide change and then boom, full, everything is gone, full genetic disorder has happened. So a classic example of point mutation is the change of a single base pair in a gene for beta globulin chain that results in the change of amino acid residue, that is from glutamate to valine. It results in disease condition called sickle cell anemia. Effect point mutation that inserts or deletes a base in a structural gene can be understood by following simple examples. So this simple example is going to be Ram has red cap. Okay, Ram has red cap. I think all of you are following. Okay. Yes, Ram has red cap. Now Ram has red cap sounds very decent, nice. Okay, ma'am, I understand totally. The words are matching. It means fine. But imagine if letter B. Okay, here what they have done. This Ram has red cap. They added a letter here. Can you see? They added a letter B here. When they added the letter, we told you that the genetic code is going to be in a continuous fashion. Yes or no? Genetic code is going to be read in a continuous fashion. If it is going to be read in a if it is going to be read in a continuous fashion, do you think the tRNA or the ribosomes are going to understand that, okay, first it was Ram has red cap, I think now they installed a B? No, they're going to keep reading it like this only. That is, Ram has DCAP, like this only they'll give, and P will be left out. Same thing if we insert one more letter. See, now they've inserted I also. Okay, they inserted I also. Now Ram has B, EDC, AP. That means again it is going to read the 3 codons. It's going to read the 3 codons and it is going to correctly, like, this is an example, okay? All this is not there, but just I'm telling you this is an example. So imagine they'll just keep coding. Okay, then we added one more letter. See, now Ram has big red cap. Makes total sense. Okay, statements could be read. That is what happens, students, by deleting, okay, by adding the first, let me say adding, by adding a single letter. Okay, single letter means I'm talking about nucleotide in your this. Okay, single letter, it can change, okay, it can change my reading frame. Yes or no? I was comfortably able to read that Ram has red cap, but now I'm not able to read it anymore. Why? Because my frame shift happened. Okay, the reading frame shift happened. Same way, it can change the, or it can cause, okay, it can cause frame shift mutation. Frame shift mutation. Yes or no? So when I am reading something, okay, now let's take Tanuja. Okay, Tanuja. Now Tanuja's name is in such a way that there is 3 codons. Okay, there are 3 codons. But the moment I add a letter to Tanuja's name, okay, Tanuja. Imagine I added an I here. Okay, I added an I here. Now what is going to happen? I'm not able to comfortably going to read this name because it's going to be Tan, Uj and A separate. So that my reading frame has shifted. Okay, again I go on to add. Let's say this is a name, so obviously we cannot land up in a good reading frame. But I'm just telling you, see, if I add a J, if I add a J, again it keeps changing. Yes or no? It keeps changing. Okay, so this will keep happening. So same thing, students, if I add a letter, I should make sure that I am adding up to 3 letters. I'm adding up to 3 letters. If I'm adding up to 3 letters, then my reading frame will not change. Okay, my reading frame will not change a lot, because why? That is going to make one codon. So same way, if I'm going to delete also, the same rule goes. That is, if I'm deleting, ma'am, you cannot delete 1 letter. You cannot delete 2 letters. You have to end up deleting 3 letters. So the reading frame shift will be proper. Okay, reading frame shift will be proper. So Ram has EDCAP. So what has happened here? Letter has been, we start deleting it. So here, all these letters are there. Now we start slowly removing. E got removed. Okay, next D will get removed. Okay, then C will not get removed. So these 2 letters we removed. So it ended up having Ram has cap, which is okay, again okay. Understood? It's again okay. So this is the thing that we need to focus on. When we add, we should add up to 3 letters. When we delete, we should delete up to 3 letters. This will cause your frame shift mutation or deletion mutation. So if we are adding, it will lead to frame shift mutation because we are not able to read it properly, right? The entire frame is getting shifted. If we are going to delete, it will lead to deletion mutation. So insertion or deletion of 3 or multiple bases, insert or delete in 1 or multiple codon, hence 1 or multiple amino acids, and reading frame remains unaltered. So what here they're trying to say, it's like when puzzle they have given it. So they're trying to say, if you're adding, you have to add multiples of 3. Okay, multiples of 3. Okay, then the reading frame will not change. Reading frame remains unchanged. Okay, remains unchanged. Clear? Chalo, okay. Now, I told you that this mRNA has codons. Yes or no? I wish I could copy paste, students. So many times I have to write same thing, but I cannot copy paste. Sad reality. Okay, this is one code. Okay, this is my mRNA. Let's say this is from 5' to 3'. Now, codons are there. Who is going to read the codons? mRNA is the one who has to get translated. mRNA has to form protein. Can mRNA read its own codon? Okay, can mRNA read its own codon and be like, oh, AUG, U code for methionine, U code for phenylalanine? It cannot do that. It does not have the ability. So that is where RNA is going to come in help. RNA will be like, don't worry, I have something called as an adapter molecule. Okay, RNA will be like, I have something called as an adapter molecule. Okay, adapter molecule who is going to do the job of reading. Okay, who is going to do the job of reading. Okay, so this reading frame, okay, this reading frame is going to happen because of your tRNA. Okay, because of your tRNA, and it is going to read these amino acids. Okay, it is going to read these amino acids and code for one thing. Okay, now I don't know how I've drawn it, but then you understand, no? Okay, from a very beginning of the proposition of code, okay, from the very beginning of the proposition of code, it was clear that Francis Crick that there has been a mechanism to read the code. Okay, and also link it to the amino acids, because amino acids have no structural specialties to read the code uniquely. I told you, no, mRNA does not have anything. Even amino acids, who is like, okay, they'll be like, AUG is here, and one amino acid will tell another amino acid, see, I am AUG, I will code for methionine. That fellow will have to code for phenylalanine. They cannot talk like how Nishant is talking to me, how Tanuja is talking to me. They cannot talk, right? They don't know that, okay, I have to code for this person. So such a mechanism does not exist. So that time what we have to do, we have to call a person from outside. We have to tell, you come and please fix this and give. So tRNA will come and it will act as an adapter molecule where you have your mRNA strand, and it is going to read the strand and be like, AUG, U code for methionine, AAU, U code for this. So that will continue. Understood? That will continue. So he postulated the presence of an adapter molecule that would one hand read the code as well as another hand would bind the specific amino acid. I am telling you that, ma'am, it is going to read the code, but who is going to add the amino acid? He is only going to add the amino acid. And who is that person? It's going to be a tRNA. So all of you look here with this diagram only, I can easily explain it to you. This is the mRNA. I told you, no, this is the mRNA. Now see, A, G, U is there on the mRNA. Now tRNA, this is the tRNA that you are going to see. This tRNA has to start reading it and tell that who is going to code for who. So here AGU, first thing tRNA is going to do, tRNA has an anticodon loop. Okay, it has an anticodon loop and it has an amino acid acceptor site. Okay, amino acid. Okay, amino acid site. So here he will code, he will do anticodon. Okay, he'll do anticodon and then he will code for it. So look here, he has done A for U, G for C, U for A, anticodon he has done. Now as soon as UCA is done, this people will understand that it is coding for serine. Okay, it is coding for serine, they will attach the serine. Again, as we go further, he'll see UAC. So he'll do AUG. Then he'll understand, okay, you have to code for, your UAC will code for your type. So like that, they are going to understand each of the codons and add and continue and form a polypeptide chain. Is this clear to all of you? Clear to all of you? So students, here, look here. Okay, let's again, I don't know how many times I'll have to draw mRNA today. Okay, I've just kept this like this. Okay, now students, see tRNA. Okay, this tRNA will not come alone. Okay, this tRNA, okay, this is the tRNA. I'm trying my level best to draw with this pen. Okay. This looks so bad, but adjust, students, please adjust. Okay, so let's say this fellow will not come alone. What he'll do, no, he will have, so this guy who the tRNA is going to have, or is going to form a combination which will have a small ribosome unit. Okay, small ribosome subunit and a large ribosome subunit. Okay, one larger subunit and one smaller subunit. It could be ATS. Okay, and ATS is not like you do this, but understand 50S and 20S like that. Like the calculation is not because of the numbers, it is because of the Swedberg's unit. Okay, now students, look here, this fellow who is the small unit and subunit, so the small unit will come and attach, only the large unit will come and attach later. Okay, small ribosome will come and attach, then the large ribosome will come and attach. Okay, so these are the ribosomes. I draw an ice, it will look like one cute thing, but yeah, this is the smaller and upper subunit. So now these two people will come. Okay, this is the larger subunit, this is the smaller subunit. They, with the tRNA, is going to read the codon. So as and when they are reading the codon, students, remember that here one amino acid will attach, then another amino acid will attach, okay, then another amino acid will attach. So like this, one amino acid will come, read the code, attach, read the code, attach, read the code, attach, and keep continuing. Okay, I'll show you the system. So this remember that the reading people are going to be your ribosome plus tRNA. Is this clear to all of you? Ribosome plus tRNA is going to code for this. All of you, I hope it is clear. So how they are going to code now? This is the amino acid. Exactly, it looks like that, no? This is the amino acid, like this they are going to code. They're going to read like this. Okay, and on this portion, who tell, on the ribosome you're going to have 3 sites. What are the 3 sites? E site, P site, A site. Yes, E, P, A site, correct. So this is going to be the exit site. This is going to be the peptide site where the chain is going to be formed, and this is going to be the acceptor site. That is, students, very simple. One fellow is going to come, one tRNA is going to come and it is going to code. After that, it is going to attach here. Here the polypeptide chain will be formed and it is going to exit from here. One is coming, joining, going. One is coming, joining, going. That's it. Okay, like our class, one is joining, coming, going. Okay, this is only what is happening in our class also. See, translation. Translation refers to the process of polymerization of amino acids. Okay, polymerization of amino acids to form a polypeptide chain. That is the goal of translation. That is, ma'am, very simple. I have to form from mRNA, I have to form proteins. But to form proteins, I do not have anything that will read the codons that is on my body. Right, so the mRNA will be like, I have codons, but I cannot read it. So what I'm going to do, I'm going to take the help of tRNA plus a ribosome. And I'm going to read and form the polypeptide chain. Understood the cycle? Chalo. Now the order and the sequence of amino acid are defined by the sequence of bases in the mRNA. The amino acids are joined by a bond called as peptide bond. Okay, so the one amino acid is going to attach to another amino acid, going to attach to another amino acids. This is going to happen because of a peptide chain. Understood? Peptide bond. Okay, now formation of a peptide bond requires energy. Formation of any bond for that matter requires energy. Okay, therefore in the first phase itself, amino acids are activated in the presence of ATP. So first one, charging process will happen. Okay, that is called as charging of tRNA. What like charging means, where the amino acid will come and bind and they will charge the tRNA. So linked to the cognate tRNA, a process commonly called as charging of tRNA or amino acylation of tRNA, to be more specific. If two such charged tRNAs are brought close enough, the formation of peptide bond between them will happen. So students, first, okay, so first what is going to happen? Very simple, we are going to charge the tRNA. We are telling that you are going for a big war, let me charge you. So let's give you food, you know, water, milk and all of that. No, like that charging of tRNA will happen. This charging of tRNA is going to be the first step, or you remember it as amino acylation. Amino acylation. See, for NEET students, we teach a little more. We write one equation and all, but don't need that much for you guys. So here, this is the first step where you charge the tRNA. After charging the tRNA, imagine this is also the charged tRNA. I don't know how to draw an easy tRNA. Okay, let's say this is the tRNA. This is one tRNA, and this is going to be, yes, Harsha, and your messages are visible. Okay, this is another tRNA. Now, let's say Tanuja has given charge to this and Harshain has given charge to this. Now when these two are coming together, automatically it is going to favor them for forming the peptide bonds. Okay, it's going to favor them to form the bonds. Like imagine Tanuja also is like a very serious NEET aspirant. Harshan also is a very serious NEET aspirant. So whenever they meet, they will always have conversations about how studies is happening, how this is happening, how that is happening. So it's going to automatically make a bond between them that is going to be so academical and so, you know, like peaceful, because they're going to talk about studies and they're going to have a like a conversation that is going to favor. Like same way, this fellow also is charged, this fellow also is charged. So both of them, when they come together, it will automatically favor energetically. Peptide bond will be formed. The presence of catalyst would enhance the rate of peptide bond formation. Okay, they're going to be catalyst which is going to be present, that is going to favor. Okay, that is going to favor the you know, peptide bond formation. Now listen, very important. The cellular factory responsible for synthesizing protein is ribosome. Okay, the ribosome consists of a structural RNAs and it's about 80 different proteins. In its inactive state, it exists as two subunits, a large subunit and a small subunit. When the small subunit encounters an mRNA, the process of translation of mRNA to protein begins. This is very, very important. Okay, this is very, very important. They are telling that when the small, you know, ribosome will meet the mRNA, okay, will come and meet the mRNA, you know, or encounter the mRNA, the entire process will happen. That means the tRNA is going to bring the larger subunit and help in the joining. Okay, the tRNA will be like, you come with me, I will take you, and the tRNA and the larger subunit will come and join. Then when the smaller subunit comes and encounters the mRNA, automatically translation is going to happen. This is a very important question. I hope all of you are making note in your NCERT, because students, you will have a tendency of not reading it properly. So when you, like, you know, you're reading suddenly, okay, when small unit encounters the process of translation, okay, these are the two steps. So when you're reading like this, you know that you did not focus on the word that smaller subunit has to come and code with mRNA for the translation to happen. You did not listen to that. Okay, so make a note of this. Now there are two sites in the large subunit for subsequent amino acids to bind and thus be close enough to each other for the formation of peptide bond. Now see, the ribosome also act as a catalyst, that is 23S RNA in a bacteria is an enzyme called ribozyme. Okay, all of us know the enzyme is called ribozyme. Okay, it can act as an enzyme and it is seen in your bacteria for the formation of peptide bond. So you can write this is going to be formed as a peptide bond. Usually we do not teach all this, okay, because we rarely see questions out of this. A translational unit in mRNA is the sequence of RNA that is flanked by the start codon and a stop codon and codes for a polypeptide chain. So students, if translation has to happen, okay, translation has to happen, then we have to make sure that this translation unit in mRNA is going to sequence, that it's going to read the RNA and it is going to have a start codon. Okay, AUG is the start codon. He'll be coming like this. Okay, let's say let's just imagine this is Harshain. Okay, this is Harshain and he's coming with a lens. Okay, this is the lens in his hand. He's coming with a lens like this. Okay, and some codons we have written here, something we have written here. Now Harshain is going to look. Okay, he's going to look for the codon. He's very sad, so he's going to look for the codon like this. Okay, read. He does not know when to stop. So here what is going to happen? Here Tanuja is going to stand and she's going to show the stop sign. Okay, she's going to show the stop sign, telling stop. Okay, stop. So he's going to read. No, now this is human nature. That okay, Harshain is coming and there Tanuja is standing. Let's say Tanuja is the police and Tanuja has a stop sign. So Harshain is going to read like this, and he's going to stop. But that cannot happen here, no? So what is going to happen? In the mRNA only, last May they will give you that this is the stop codon. So when the tRNA is reading, binding and forming the peptide chain, it will reach UAG and it will just stop. Otherwise the polypeptide chain will keep becoming longer and longer if there was no stop codon. Yes or no? So this will keep continuing. Understood? This will keep continuing. So this is what they're trying to say, and now they're trying to say that polypeptide chain. And mRNA also has some additional sequence that are not translated, are referred to as untranslated regions. Okay, the UTRs are present in at both 5' and 3'. 5' they have it before. Now here, how did Harshine start? Because I told start. Okay, I told start Harshine. Okay, start reading. So that start sign was AUG. Before AUG, okay, before AUG, there was one more set of motivation that he had to start. That was because of your UTRs. That is untranslated regions. These untranslated regions are present to increase the efficiency of translation. Okay, so that stop happens, start happens, everything is going proper. We will start this. Got it? Okay. Harshine is boy only, no? Because you people put such fancy names, it's so difficult to understand. Then later they'll be like, ma'am, you change my gender. Okay? You should, like, reading some names, I think that, okay, this could be a male or this could be a female, and then suddenly they'll be like, ma'am, I am not, I'm a girl, I'm a she, I'm a he. So you people should tell me. Okay, you're reading your name, I cannot decode because I'm not tRNA, I'm not an adapter molecule. I cannot decode. Girl, see, problematic it is. I should stop taking names. So we will draw hair for her because she'll feel bad. Okay, got it. Yes. Chalo. Now, they are required for efficient translation. For initiation, the ribosome binds to the mRNA at the start codon and it is recognized only by the initiator tRNA. The ribosome proceeds to the elongation phase of the protein synthesis. During this stage, complexes composed of an amino acid linked to the tRNA sequentially bind to the appropriate codon in mRNA by forming complementary base pairs with tRNA codon. I don't know why they have complicated it. It's very simple. They are trying to say that as and when you form, your anticodons will be formed and later you will keep adding your polypeptide chain. The ribosome moves from codon to codon along the mRNA. Amino acids are added one by one, translated into a polypeptide sequence dictated by DNA and represented by the mRNA. At the end, a release factor binds to the stop codon, terminating translation, releasing the complete polypeptide chain. So students, after the this also, what is that? So this will look like this. Okay, this is the thing here. Okay, here UTR is there, untranslated region. Okay, untranslated region is there. Then here who is there? I am there, telling her AUG. Okay, start. So here something is going to start. Okay, something is going to start. Then it will keep continuing, and here there is going to be stop. Okay, that is, let's say UAG is there to stop. After that, who is going to be there? UTR is going to be there. Understood? Other than that, one more person is going to be there. That is going to be the release factor. All these people are going to help in termination. See, UTR is mainly present for efficiency, students. Okay, UTR is mainly present for efficiency. But this is going to be start. Okay, and this is going to be stop. These people are having fun. They don't even know. They're just pranking me, I feel. Who didn't mark the attendance? Quickly mark the attendance. So students, I couldn't take the class last time on NEET English was because of my eye checkup. So I have got an infection inside my eyes. I have a pimple which has become infectious because of too much light. Okay, light, because the studio lights. So tomorrow I'll take the biotechnology part 2 class. Okay, so now what happens is I have to usually, like, after, like, when my eye starts hurting, I have to take a break and I just have to close my eyes and just sit for 10 minutes. Okay, so I think it's going to be a little hard task tomorrow because there's going to be a lot of lights, and I have to, after every 1.5 hour, I should take a 10 minutes break. So yeah, it's a little sad, but yeah, I got it. Like, I have to get the glasses, one more glasses I have to get. So that was the thing. Yeah, this is the blue light one, but they're telling me I have to get a better version of it. Okay, a better version of it because of too much strain to the eyes. So as I told you guys, there is a lot of classes that we are taking nowadays. So because of which screen time has become more. Like you make the notes and then you sit and prepare. So I study also from my own videos. So I study from laptop. So again laptop, again laptop, again laptop, then big screen. So it's throughout the day I don't get rest for my eyes. So because of that, I have created a problem in my life. So it will take some time, I think, to rectify. So yeah, what to do, students, what to do? So many problems. Our body is very sensitive, and even like I've never done it, so I've not taken this much of screen time. So since I have not taken so much of screen time, the moment it started changing, and from past 2 months when I started having increased screen time, automatically the pimple has come, and the pimple has gone down, but it has become little infectious. So after some time I start getting here pain and all of that. Okay, so I'm telling only you guys. Yeah, that's the problem. Now you guys also will be facing it because of your classes and everything. It's actually very harmful. But what to do? Everything has become online that we do not have an option now. Okay. So this glass is Blu-rays, but this is I think not enough. So they're going to give me another one. So I have to use that and teach. So whenever someone asks in the class, you guys can give the explanation, because since you guys are here, I thought I'll tell you. Okay, because a lot of students are asking me, ma'am, what happened, and they were not able to contact me. Yes, okay, cool. So students, this is clear, no? So this, don't forget, UTR is going to be present before the start codon and after. Okay, after the stop codon. And upon that, they're going to have release factors that is going to help in the termination. That is, they're going to tell this fellow that stop, don't come. So like, don't read anymore. It's enough. Okay. And these people, how they're going to go? They are going to go from E side to P side to A side. So one is going to come here. Okay. Next one is going to come here and form the first polypeptide. Then in the next one, another fellow will be waiting to come, and this fellow will be exiting, and here there will be one polypeptide. So like this, it is going to form chain. I don't know if I added the diagram from NCERT. If I added the diagram, I'll show you guys. Wait, let me try to open NCERT, students. I think I have the NCERT here only. Wow, it's like so many blind people in the class, teacher and students. I told about my eye problem. They're like, ma'am, we also have so many eye. We already have eye problem. Yeah. Wait, students, once again, I'm just showing the screen. Okay. I forgot to add this image. So that's fine. Yes, 1 second. I'll show you guys the image. Yes, can you see the image? Right, you can see the image, no? So here, students, what is going to happen? You can only see my cursor, if I believe. So here, you can see that these are the sides. Okay, this is the exit side, the one that is leaving. No, can you see my cursor? Yeah. So this is the exit side. So from here, it's going to exit. The one who already formed the anticodon and made the join, he's going to exit, and the next person is going to come and join. So here you can see one fellow is waiting. Okay, one fellow is waiting to come. Is my cursor seen? Yeah. Okay, one fellow is waiting to come, then this fellow is coming, this fellow is going to move. So what is going to happen? This guy is going to give this entire chain here. Now when this guy joins, this chain is going to come to him. So this is going to keep continuing. So here, this portion is the exit. Okay, this was the exit, this is the entry. So here he'll come, he'll make the anticodon, he'll join here. Now this fellow is going to exit next. Okay, he's going to exit because he already joined the alanine. Now this guy will go and join with this valine, and this fellow will come here. Now this valine with the entire chain is going to pass on. So it's like, you know, passing the ball. How we do passing the ball, no, like that only. One person will come, from there another person will come, and it will keep continuing, okay, passing the ball. Okay. Chalo. Done? Dusted? Okay. Now regulation of gene expression. What is regulation of gene expression, students? That is, regulation of gene expression means the gene can decide when I want to switch off, when I want to get switched on. Okay, when I want to switch on and when I want to get switched off. So when the gene is deciding ki, okay, I don't want to get activated now, I don't want to work now, I don't want to be part of this, I'm going to get switched off. When gene decides to do that, the gene can do that. Okay, gene actually can do that. It's not like you know, not like us, like we want to take a break, we cannot take a break until we have permissions of everyone, right? If we are working, then we need to take permission from the above people, we have to take permission from uski above people. Now if you are at school, you have to take permission from your teacher, principal. So the cycle keeps continuing, but that is not the case here. If the gene has decided to switch off or switch on, okay, if the gene has decided to switch off, okay, or switch on, then what is going to happen? The gene will be like, okay, I have decided to switch off. That means I do not want to get into this process. When can the gene do this? The gene can do this when it is at the translation phase, transcription phase. That is, before going for transcription, the gene will be like, I don't want to do this. Okay, I don't want to do this. I'll just switch off. Then the gene can decide to do this when we are doing your RNA processing. This is in eukaryotes, okay? RNA processing, yes or no? Where we do splicing and capping. Splicing. When we do splicing, the gene can decide to switch off. Next, the gene can switch off when there is a transport of your formed mRNA. Okay, formed mRNA is being transported to the cytoplasm. Okay, cytoplasm. That time also we can do this. Then or else we can do it when the gene is going for translation. Okay, when the gene is going for translation. Understood, all of you? So you can do this. What you can do, the gene can switch off when it is going to start with transcription. Okay, transcription is going to start. That time it can do. Yes or no? Okay, yes or no? Who is this? IITN. Thank you. When the gene is going to switch off, it can decide when it is going to happen in the transcription state, or when it is going to happen in the RNA processing state, or when the mRNA is going to be transported from the nucleus to the cytoplasm, or when it is going for translation. Any doubts, students? Any doubts? No doubts, no? This is the thing. So now, ma'am, why should the gene switch off? Why can't it work like us 24/7? No, we are not working, we are sleeping. No, the student's gene has decided to switch off because you do not need a gene. Okay, IIT and understood. Thank you so much. Do not spam. Okay, so students, sorry, I'm telling IIT. So students, the gene is like, I do not have to be regulated all the time. Yes or no? Why should it be going through a regulation all the time? It does not have to. Now my hand gene, oh my god, my hand gene doesn't have to be activated on my face. My face gene doesn't have to be activated on my hand. So like that, okay, like that, up and down. So same way, gene decided to switch off here. Okay, gene decided to switch off here. Gene decided to switch off here. Gene decided to switch off here. Any of these times they can switch off. But again, there's a lot of loopholes for that. Look here. See. Considering that gene expression results in the formation of polypeptide, it can be regulated at several levels. That is, in eukaryotes, the regulation could be exerted at transcriptional level, that is formation of the primary transcript. At the processing level, where there is regulation of splicing, transport of mRNA from nucleus to cytoplasm, or the translational level. Understood all of you? Yes or no? Now, see, there are a lot of mechanisms. You do not have to study all of them. You are going to study in eukaryotes, because students, prokaryotes, you're going to study in prokaryotes because eukaryotes is very difficult. Okay, like the whole thing process is going to be complex. See, the genes in a cell are expressed to perform a particular function or a set of functions. For example, if an enzyme called beta-galactosidase is synthesized by E. coli, it is used to catalyze the hydrolysis of disaccharide. So before I go into this, understood IIT, and please, you go to IIT and study. What are you doing here? Okay, IITians don't have time to waste like this, to spam like this. Chalo, look here. So students, I don't know how I'll draw a straight line. The biggest task I've always felt in my life, to draw straight lines. Okay, now look here. 1, 2, 3, 4, 5, 6, 7. Okay, 5, 6, 7. Now students, this actually drawing has gone little Pipo Z Y A. Now this here you are studying only one gene regulation, that is going to be lac operon. Okay, only one gene regulation, that is called as lac operon. Lac operon concept is for prokaryotes, that is whenever lactose is present, the gene decides to switch on or switch off depending on that. Okay, now here Pipo ZYA, students, remember there are regulatory genes. Okay, there are regulatory genes who is going to get regulated. Okay, who is going to get regulated. So now look here, what we are going to learn is very simple. First I'll teach you the process, then I'll teach you the explanation for it. See, this I gene, okay, this I gene is actually going to inhibiting. Okay, it's not like, it's not going to like induce, it's going to inhibit. Okay, this I gene is going to form repressor mRNA. What is going to form? Repressor mRNA. mRNA is going to form what? Protein. Repressor protein. Now this repressor protein has a problem. He's like, I need to go and block the road. So this repressor protein will go all the way from here and block the operator. Block who? The operator. This operator has got blocked. Now imagine transcription. For transcription, your RNA polymerase is coming. Okay, RNA polymerase is coming happily. Okay, I'll do transcription today and I will form the mRNA. All this he's telling, and he's coming happily, but he notices that there is a big block here. Now what is going to happen? Transcription will not happen. Transcription will not happen. At the same time, the gene has got switched off. Gene is switched off. So this repressor protein is going to bind with the operator and make a complete block there. Okay, because of which transcription will not happen. So here you can understand that the gene has decided to get switched off during the transcription, and here the gene got switched off. Now students, so because the gene got switched off, the ZYA also is not going to work. That is lac Z, lac Y and lac A. They are also not going to work. Okay, they are also not going to work. Then ma'am, how the gene is going to get switched on? So students, very simple. E. coli is the E. coli is the bacteria that we have taken. So in E. coli, if glucose, okay, glucose is more. You tell me, if in your body glucose is more, do you need lactose from outside? Do you need lactose from outside? It's a question to all of you. I want all of you to answer. Do you need lactose from outside? Yes or no? Okay, you have glucose. Do you need lactose from outside? Yes or no question. Okay, fast, fast. Next students are typing. No. Okay, no. Someone has Tanuja has told no. Okay, I agree with Tanuja. So this is your E. coli. Okay, this is your E. coli. Now this E. coli has enough glucose in the body. It will not require lactose. It does not want lactose. So here, when lactose is not at all required, what is going to happen? E. coli will decide that let the gene get switched off. Let the gene get switched off. Correct? Now at the same time, imagine the E. coli has used up all the, okay, E. coli has used up all the glucose. Okay, and there is no more glucose. Do I need lactose that time, students? Do I need lactose? Answer, all of you. Do I need lactose that time? Jaldi, jaldi you should answer. Yes, I need lactose. Yes or no? I need lactose. Okay. So because all the glucose got used up, so that's then my imagine I ate food and I did not eat food for around 12 hours, 13 hours. My body glucose, everything has got decreased. Automatically, what is going to happen? I am going to feel dizzy. I am going to faint. Okay, so that time, what is going to happen? I am going to go to a hospital and they're going to give me drips, right? So that means externally, I've taken the energy that I needed. Same way here also, lactose is required. That time, what is going to happen? The E. coli is like, let the gene get switched on. Okay, the gene gets switched on. Correct? Now, when the gene gets switched on, what is going to happen? You have lac Z, lac Y, and lac A. Correct? These are the regulatory proteins. That is, the I gene has to give them the permission to get regulated. So here, lac Z is going to release beta enzyme. Okay, enzyme beta galactosidase. What is it going to release? Beta galactosidase. That will do the job of breaking the lactose to glucose and galactose. Glucose and galactose. This is the job of this fellow. Y is going to give out permease. That is going to allow the permeability or increase the permeability of the E. coli cell. Increase the permeability of the E. coli cells. Then you have lac A, who is going to release transacetylase, which is actually going to increase the efficiency of beta galactosidase, but in your NCERT nothing is given. Okay, in your NCERT nothing is given. So here no function as such. Okay, no function as such. So students, these people have to get regulated, yes or no? So what they're going to do, now we will have a question. Ma'am, here lactose permeability is going to be increased when lactose already entered into the cell. Students, remember that this is a leaky model and lactose has already entered the E. coli cell for this regulation to happen. Okay, if lactose does not enter the E. coli cells, then there is no way that the gene is going to get switched on. All of you agree this, no? All of you agree this, no? So, let's go full down, okay? I have not left the slides here. So I'm going full down, then you have to come up again. Okay, here again. Okay. Okay, students, here we'll write, okay? So students, what is exactly going to happen? That you understood, okay? That you understood. Now, same thing we have to write. Pipo Z Y A, okay? Pipo Z, first I should draw a box. I have a disability of not able to draw a box. Okay, not bad. This is a good box. How many of you cannot draw a box even with scale? I am one among them. Okay, Pipo Z Y. Okay, ZYA. So students, what is going to happen here? Pipo ZYA is there. Now this I gene is going to do the same thing. He's going to form the repressor, mRNA. Okay, repressor, mRNA. This repressor mRNA is going to code for protein. Okay, do any of you have that disability? Please tell me. Okay, I think Tanuja and I have almost all the disabilities that I am telling. She's agreeing for all of it. So I cannot even draw a straight line with scale, but sometimes when I draw without scale also it comes perfect. So like that, yeah. So yes students, so now this repressor protein will go and try to bind with the operator, but since our gene has already, our E. coli already has lactose, what is going to happen? We have to make sure that the gene is going to get switched on, because there's no glucose, we need lactose. So that lactose will go and bind with this repressor protein in such a way. Okay, bind with the repressor protein. So we call this lactose as inducer. Okay, we call this lactose as inducer and we call this protein as repressor protein. So this inducer and repressor will form a complex. Okay, which will form a complex, because of which this will not go and bind with the operator. Now you tell me, who is the happiest person here? Who is the happiest person here? It's going to be RNA polymerase. RNA polymerase will dance and come, and he'll happily do the transcription, and by the time he reaches here, beta galactose is going to get released. Okay, beta galactosidase is going to get released. Okay, Y gene is going to release permease. This is going to release transacetylase. Okay, transacetylase. And finally, what is going to happen? Here obviously breakdown is going to happen. Okay, here glucose, galactose, here permeability is going to happen. Here function is there, efficiency will be increased. So students, this way what is going to happen, students? This way what is going to happen? The gene is going to get switched on. Okay, gene switch ons. Clear all of you? Clear all of you this lac operon concept? Exactly. Yes, RNA polymerase is going to be happy. Now students, understood, gene got switched on. Okay, gene got switched on. My handwriting is coming actually better, but the problem is I have reduced the size, so it is coming like long. Okay, gene has got switched on. So this is your lac operon. Now I'll go back there, and it was done by Jacob and Monod. Okay, I keep forgetting about these two people. I think they'll be mad at me for that, not always forgetting. Jacob and Monod. Yes, let's come here. Okay, so here what is going to happen, students? See, beta galactosidase synthesized E. coli. It is used to catalyze the hydrolysis of a disaccharide, lactose into galactose and glucose. The bacteria use them as a source of energy. Hence, if the bacteria do not have lactose around them to be utilized for energy source, they would no longer require the synthesis of an enzyme, beta galactosidase. Therefore, in simple terms, it is metabolically, physiologically, or environmental conditions that regulate the expression of gene. The development and differentiation of an embryo into an adult organism are also a result of coordinated regulation of expression of several genes. Okay, so you might have a question, no ma'am, why does this gene regulation happen? So you should understand, from the time we are a fetus up to the time we are becoming an adult, some genes switch off purposefully, some genes switch on, so that correct activities will happen in the body. Now in prokaryotes, control of rate of transcriptional initiation is predominant site for control of gene expression. That means transcription is the initial place where this can happen, and this is a PYQ. Okay, this is a PYQ. If you have ever seen it, you will know that this is the PYQ. In transcription unit, the activity of RNA polymerase at the given promoter is in turn regulated by the interaction with accessory protons, which affects its ability to recognize the start sites. These regulator proteins can act both positively, that is as activators, and negatively as repressors. Okay, the accessibility of promoter region of the of prokaryotic DNA in many cases regulated by the interaction of protein with sequence termed as operator. So students, the O region where this is going to go and bind, that is called the operator. Okay, and the P region is going to be the promoter, where the RNA polymerase will come and bind to start the transcription, but it will not be able to continue. Okay, yes. Tanuja, after we read this paragraph. The operator region is adjacent to the promoter elements in most operons, and most cases the sequence of the operator bind a repressor protein. Each operon has a specific operator, specific repressor. For example, lac operator is going to be lac operon and it interacts with lac repressor. So here they're just giving introduction. Okay. So here students, look here. See, I gene formed repressor mRNA. Now this repressor will go and bind with the operator, prevents the RNA polymerase from transcribing. Okay, that is in the absence of inducer means absence of lactose. Okay, here you see repressor protein is formed. This inducer, that is a lactose, will go and bind with the repressor and form a complex, and then what is going to happen? Lac mRNA will be formed and all these enzymes will be released, and then translation, transcription, everything will happen happily because your RNA polymerase will be able to come forward. Now one more paragraph. That's all students. Okay, that's all is this paragraph. I thought I added one more paragraph. Okay, this paragraph, okay, I added. Okay, human genome project and DNA fingerprinting is left. Okay, that will take us maybe an hour, so we'll come after a break. Okay. So by the time I get up and go to become 155. Okay, so we'll come back by 2:15, 2:20. Okay, it's a kuti lunch break. Okay, yes. The time will go so fast, students. Break is like by the time I go out, it'll be break. Break would be over. So see you guys. Hello students. Do you all have your lunch? Yes. Nothing special. Chalo, let's continue. Okay, we have to do HGP and your DNA fingerprinting, which I did not do in Vedantu NEET English. So those students who are watching can help themselves. Yes, ready? Guys, you're not liking, okay? I don't know why you guys are not liking nor subscribing to the channel. So much conjuicy here. So much conjuicy. Chalo. Human Genome Project. In the preceding sessions, you have learned that the sequence of bases in DNA that determines the genetic information of an organism. Right? DNA decides the genetic information of an organism. In other words, genetic makeup of an organism or an individual lies in the DNA sequence. That is, genetic makeup means what? Now, how each of us are looking, okay, how each of us are looking totally depends on our parents. Okay, it depends on our parents. It depends on one second. Okay, it depends on our parents, it depends on our genetic makeup. Right now, if a disorder is coming, even that disorder also totally it is dependent on if it is there in the genetic line. Okay, if it is there in the genetic line. So same way here also, the genetic makeup is decided by each individual, lies in their DNA. If two individuals differ, then their DNA sequence should also be different. Yes or no? Now, at least at some places. These assumptions led to a quest of finding out complete DNA sequence of a human genome. So people started having the doubt that how would it be in, twins? How would it be in fraternal twins? How would it be in two different people? How would it be from a mother to a child? So people had the quest, that means the thirst to find out what exactly happened. Okay. With the establishment of genetic engineering, which right now you're studying with me in your biotechnology techniques, where it was possible to isolate and clone any piece of DNA, and availability of a simple and a fast technique of determining the DNA sequence. Yes or no, students? Right now we have learned all this in biotechnology, where we can simple and fast technique where we can determine the DNA sequence. Yes or no? By the technique of genetic engineering. Okay, by the technique of genetic engineering. So it is possible to isolate, clone, and any piece of DNA availability, simple and fast. Okay, simple and fast. Now it's a very ambitious, HGP was a very ambitious process, project. It was not done by just one or two people, it was done by a lot of people involving, different countries were involving, and they were ready to give money for this project. Okay, so Human Genome Project. Students, this was not just because they wanted to know the sequence, it was also a big factor that could have cured a lot of genetic disorders, changed a lot of things in in the society. So that's why they all wanted to invest in this. Okay, it was a mega project. You can imagine the magnitude and the requirement for the project if we simply define the aims of the project as follows. So look here. Human genome is said to have approximately, that means our body is said to have approximately 3 into 10 to the power 9 base pairs. Okay, and if the cost of sequencing required is $3 per base pair, the cost estimated in the beginning, the total estimated cost of the project would be approximately $9 billion US dollars. Students, US dollars 9 billion. Okay, so you can convert it in Indian and check how much it is. It is going to be a lot, right? A lot. So further, if obtained sequence was stored in a type of form of book. Now, okay, ma'am, we decoded the sequence. You people were a part of the mega project. You calculated in such a way that, okay, DNA around 3.3 into 10 to the power 9 base pair is there in human, human genome. Then each of them will be calculated as 3. It will cost us around 9 billion. Okay, it will cost us around 9 billion. Now, after doing that, now we have to store this information and keep. Now, where will we go find a, imagine each individual has a book. Okay, each individual has a book of their genome. Okay, they are haploid content of the genome 3.3 into 10 to the power 6. They have a content of each of the genome. Then how many books we would need? That is, if each page of the book contained 1000 letters and each contained 1000 pages, then 3300 such books would be required to store the information of a DNA sequence of a single human. That means me, Tanuja, each of them would have a book which had 3300 pages. I mean 3300 books would be there. Can you imagine? 3300 books, not pages, 3300 books with each of 1000 pages. Okay, it would be such a hassle to even store data of anyone like that. So then the enormous amount of data expected to be generated also necessitates the high-speed computational devices for data storage and retrieval and analysis. So HGB was closely associated with rapid development of new area in biology called as bioinformatics, where we were using biology plus computer science, okay, and trying to store and preserve, like analyze the data. Okay, store and analyze the data. Analyze the data. Got it? Yes. So bioinformatics is nowadays very common. Okay, even I have studied bioinformatics. So bioinformatics is very, like, people really love it and a lot of people go to that field also. Okay. Now, what was the goals of HGB? When they started this HGB, what was the ultimatum, or telling that, okay, what was the ultimatum or what was their goal? So see, some of the important goals of HGB were that identifying all approximately 20 to 25,000 genes. Okay. So determining, so first one was to identify all 20 to 25,000 genes in a human DNA. Determine the sequence of 3 billion chemical base pairs that make up the human DNA. Okay. Store this information in databases. Improve tools for data analysis. Transfer related technologies to other sectors such as industries that can make, medicines, that can make, like how we are going to study in biotechnology, the applications, different types of applications, be it BT cotton, Bacillus thuringiensis, or be it, ripening of your tomato, forming tomatoes, all this. Like, for that we had to send these technologies to the other industries, address the ethical, legal, social issues that might arise from the project. So all these, okay, all these were the, okay, all this were the biggest thing. Got it? Yes or no? Put the stucker. Chalo. Yes. So the goals of HGB was identifying all approximately 20 to 25,000 genes in human DNA, determining the sequence of 3 billion chemical base pairs. Okay. Store this information in databases, improve the tools for data analysis, transfer the related technologies to other sector, and ethical, legal, social issues. LC. Okay, LC, you might think like, oh my god, my aunt's name is LC, but LC's full form they will ask you. Okay, LC's full form they will ask you. Full form is important. Students, goals of HGB, salient features of HGB is very important. Okay, very important. See, besides providing clues to understanding human biology, got stuck. Yeah, human biology, learning about non-human organisms DNA sequence can lead to an understanding their natural abilities that can apply towards solving challenges in healthcare, agriculture, energy production, environmental remediation. That's what I told you. It was not just helping in telling that okay, I will just store this data and keep on storing this data, I will just like spend a lot of money where different countries are coming in and I'll spend a lot of money. That was not the thing. It was like to understand how much like now decide recombinant DNA technology. All this is because of knowing the sequence, right? Otherwise, how would we do it? How would we know that there is an ORI site? How would we know there are tetracyclines, ampicillin, there are selectable markers? All this information comes from this HGP. So HGP is actually a big project, right? So many non-human model organisms such as bacteria, yeast, elegans, that is a free-living non-pathogenic nematode, Drosophila, plants, etc, etc, etc. Okay, so all these came from a place where people were able to identify. Okay, got it? So HGP was not a baby project that you can think of. It is a very, very mega project. Now students, there are some methodologies. Okay. Methodology. So two techniques were used during HGP project. The first one was, the method involved two major approach. Two approach was there. One was the approach which was one was a blind approach and one was a more selective approach. One approach focused on identifying all genes that are expressed in RNA. That is, whoever is expressing in RNA to form the protein, whoever is going to code for protein, we will take all the genes and we will call it as expressed sequence tags. Okay, ESTs. Now the other was a blind approach. Okay, other was a blind approach of simply sequencing the whole set of genome and contained all the coding and non-coding sequence. That is, they took an entire genome. Okay, they took an entire genome, all the genomes, they took, be it introns, be it exons, they took all the sequence, and this was a blind approach. So they took a blind approach of simply sequencing the whole set of genome that contain coding and non-coding, and later assigning different regions in the sequence with a function which is termed as sequence annotation. For sequencing, the total DNA from a cell is isolated, converted into random fragments of relatively smaller size. Okay, so recall, DNA is a very long polymer and there is a technical limitation in sequencing a very long piece. So we will make it into kuti piece. Okay, and cloned and suitable host using a specialized vector. Students, so those who studied biotechnology, this will be so easy. Okay, see, there are technical limitations and cloned in suitable host using a specialized vector. The cloning resulted in amplification of each pieces of DNA fragments. Okay, amplification means what? We increase the number. We can do that with the help of PCR as well. So here we did amplification, that is sequentially that could be sequenced with these. Now the commonly used host were bacteria and yeast, and the commonly used vector were BAC and YAC, which I told you in a previous class as well. BAC and YAC. 1 second, students. So here they're telling that what was the methodology. Methodology is important. They'll ask you the full form also. Oh God, what is the clearly, no? Okay, methodologies. So students, something is wrong with the spelling. What is the spelling? Yes, methodologies. So methodologies mainly had two pathways. Okay, two pathways were there. What is the two pathway? One was expressed sequence tags, ESTs. That means what? Only the expressed one, who was expressing itself to form the genome. Okay, who was expressing itself to form the expressing itself in the RNA. Okay, expressing itself in RNA was taken into one category. The next category was a blind approach. Students, they'll ask you, okay, blind approach, express sequence tag. So do not forget the full form. Okay, blind approach. Blind approach was taking all the coding plus non-coding sequence. Non-coding sequence. Okay, you know it's a bachelorette. Why will you take couples and go? Yes or no? You know it is a bachelorette, only bachelors are allowed. Why will you take a couple and go? It's like that. All coding and non-coding sequence and the whole genome. Okay, whole genome was taken into consideration. Whole genome. Okay, now after taking the whole genome, they started giving one piece and trying to label them. So that was called a sequence annotation. Sequence annotation. Okay, sequence annotation. Now after sequence annotation, what did they do? They took a small piece of DNA. Okay, they added it into a vector. Okay, that vector was added into the host, and the commonly used host was bacteria and yeast. Okay, then amplification, other things happened. Then vector was back and yak. Back and yak full form is bacterial artificial chromosome, yeast artificial chromosome. Okay, don't forget yeast artificial chromosome. So they did this, and vector was added, a small desired gene, we tell, no, like that desired gene was acted into a vector to the host. After that, amplification and other things. Now here, look at this. Okay, this is the person, this is the beautiful lady. Inside her cell we are taking the DNA. Okay, we are taking one chromosome. This is a chromosome, and this chromosome was formed because of our packaging. From that chromosome we are checking what is there in this chromosome. We will see the DNA. Okay, the DNA strands. When we take the DNA, we'll be able to see the sequences. Okay, we'll be able to see the sequences, and then later, after the sequences are seen, we'll be able to allot it. Right, allot it means what? That okay, you are sex chromosome. Okay, you are sex chromosome. Okay, your sex chromosome X. Okay, and your Y. So like this, we have to denote. No, we have to tell that these people are responsible for that. Okay, for that we had to do this. Got it? Yes, we had to do this. So this is the process. This is how it was done. Okay, now what is happening? Now sequencing. See, the fragments were sequenced using automated DNA sequencer that worked on the principle of method developed by Frederick Sanger. Okay, it was developed by Frederick Sanger, and it was called a sequencer. Okay, DNA sequencer. Okay, Frederick Sanger was the person who found this. Okay, Frederick Sanger. And Sanger is also credited for developing method for determination of amino acids. Okay, so he's like a big person. Exactly. So many ads, even I can see the ads. Okay, so yes. So students, it's time to take premium for all your accounts. So determination of amino acid sequence. These sequences were then arranged based on some overlapping regions. Okay, overlapping regions present in them. This required generation of overlapping for sequence. So students, when the sequence was there, okay, actually overlapping sequence is a very complicated topic. Okay, I tried as much as possible to find something that will connect you guys, but there's nothing. It's very complex topic. Okay, now students, imagine overlapping means, let's say these two sequences are overlapping. Okay, these two sequences are overlapping. We cannot physically remove this. Yes or no? Can we do the physical alignment telling that, oh, you are overlapping, move that side, give little place? We cannot do that, no? So what they did, the sequence were then arranged on some overlapping regions present in them. This required region of overlapping fragments for sequencing. Alignment of these sequences was not humanly possible. Okay, therefore specialized computer-based programs were developed. These sequences subsequently annotated and were assigned to each chromosome. Understood? So what we are going to do? We have taken a set of people, and right now we are going to do naming ceremony of that person. Okay, that is what is happening here. That is, we have taken a set of sequence. Okay, this is a sequence, let's say, that is going to do some job. I will align this sequence to this chromosome, telling you take up this. Same way sex chromosomes also. Okay, sex chromosomes also, we will tell them that, wait, you are going to do this, you are going to determine the sex of the baby, you are going to do this. So like that, 1 we started doing. Okay, the sequence of chromosome 1 was completed in May 2006. Students, always first does not mean you're actually first, because the example is your chromosome 1. Chromosome 1 was one of the last chromosomes to be annotated. Okay, so it was done recently in 2006. I think all of you are already born, no? Right, all of you would be born by 2006. Okay, 2006. Okay, yeah, sure, I'll do that. Okay, 2006, what is going to happen? Chromosome 1 was annotated. This class I will not give punishment. That class I'll give punishment. Biotechnology, if you people don't attend. That is so for sure. First hitting solely for you guys. Okay, so here the sequence of chromosome 1 was completed in May 2006. This was the last of the 24 chromosomes. That is 22 autosomes and X and Y. Okay, X and Y. All of you know 23 pairs and all of that. And one pair is your sex chromosomes. Now, salient features of human genome. Oh, where is this going? Salient features of human genome, students. See, salient features of human genome is that the human genome contains of 31674 million base pairs. Okay, the average gene consists of 3000 bases but vary greatly, with the largest gene known as dystrophin with 2.4 million bases. So this you please remember and keep. I know other numbers you might forget, but try to remember as much as possible. Usually they won't ask the numbers, but yeah, sometimes they ask. Okay, like dystrophin and all, they can ask. Okay, dystrophin they can ask. Now the total number of genes is estimated at 30,000, much lower than the previous estimate. Previous estimate was around 80 to 1,40,000 genes were. Okay, all the 99.9% nucleotide bases are exactly the same in all people. So students, here what they're trying to say, they are trying to say you, me, everyone are the same around 99.9%. That is, we have a bulk DNA which is present in almost all the humans. We have a bulk DNA that is present in almost all the humans, but that 0.1% is what is making you and me different. Okay, you and me different. Understood, all of you? So it is not like a mass of DNA that has changed in all the humans. 99.9% is the bulk DNA which is present in all the organisms. What has changed? A 0.1% has changed, and that 0.1% is enough to make you and me look so different, you and me behave so different, everything different. Understood? So that 99.9% DNA, you can call it as bulk DNA. Okay, bulk DNA. Now, the functions are unknown for around 50% of the discovered genes. Now, students, all the genes does not have to have functions, no, immediately. So, gene is already a small portion. Now, all the genes need not have a function. So, less than 2% of the genome codes for proteins. Can you understand? That means the central dogma where your DNA becomes mRNA and mRNA becomes protein, this is only done by a 2%. Okay, only 2%, that is less than 2% of them. So you can understand how tiny all this happening is. Repeated sequences make up a very large portion of the human genome. Okay, repetitive sequences are stretch of DNA sequences that are repeated many times, sometimes 100 to 1000 times. They are thought to have no direct coding functions. They shed light on the chromosome structure and dynamic evolution. So students, here listen carefully. Now let's take example of, whose example do we take? Let's take example of Chota Bheem. Okay, Chota Bheem. Okay, Chota Bheem and me, Gopika. Now let's say Chota Bheem has a sequence of A, G, G, A, T, T. Even I have the sequence. Okay, because I like Chota Bheem. Okay, A, T, T. It's the top secret. Don't tell anyone else, students. Yeah. Tanuja, that only we can expect from these places. Okay. So students, look here. This is one sequence and this is one sequence that both me and Chota Bheem has. But Chota Bheem has this around 10,000 times. Okay, this is repeated around 10,000 times. But for me, it is only repeated 10 times. It is repeated only 10 times. Understood? So, repetition matters. That is, they are trying to say that repetitive sequences are stretch of DNA sequence that are repeated many times, sometimes 100 to 1000, and they are thought to have no direct what is the direct coding functions, but they shed light on the structure, dynamics, and evolution. So what Chota Beam has, even I have, but for me, for Chota Beam, it's repeated 10,000 times. For me, it is repeated 10 times. But at the same time, maybe there's another sequence. Okay, there's another sequence like G, 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 C, C, T, T, A. Okay, and Chota Beam, let both of us have this sequence. Okay. Chota Beam has it repeated around 1,000 times. I have repeated around, let's say, 40,000 times. So understood. So it does not make anyone better or whatever, but it makes the change, it makes the evolution, it makes all of us different. Okay. So now students, see, chromosome 1 has most genes, that is 2968, which was also found at the last. Okay, it was found on May 2006. Now this fellow Y gene has the fewest, which is around 231. Okay, now scientists have identified about 1.4 million locations where the single base DNA difference, that is SNPs, single nucleotide polymorphism, pronounced as SNPs, occurs in humans. This information promises to evolutionize the process of finding chromosomal location for disease associated sequence and tracing human history. Okay, human history. See, so here I'll write you, what is an SNP, ma'am? Mintu ma'am has Chota Beams. Yeah, actually nice. No, Mintu Chota. Chotu. So students, here what is going to happen? They are trying to say that AGA CCT, okay, ACA CCT. Now this, let's say this G, students, this G is present in Chota Beam. This is present in Mintu. Now can you see this G to C made so much of difference? Okay, so much of difference for all of us. Understood? That is made everything different, be it from the hair texture to how we look, everything changed from this G to C. Understood? So this is called as SNPs. SNPs means what? Single nucleotide polymorphism. Okay, that is one nucleotide changed. It changed the whole polymerization. Yes, Basavasauruses. We had kept him a name, no? Pintu. Pintu. Students, you have to check that short. In that short, I'm telling his name. Okay, in one of the shorts, I'm telling his names. Pintu, I guess. Okay, is this clear? So Basavasauruses is better, so that we'll be able to remember. Yes, so single nucleotide polymorphism. Okay, single nucleotide polymorphism, that is your SNP. One nucleotide change, that nucleotide made so much difference in both of us. Okay, lot of it, be it the physical appearance, be it the texture of the hair, everything has changed. Or it could also be that both of us have straight hair, but both of our textures also are different. So that kind of changes happen because of a single nucleotide polymorphism. Oh, what is this dancing? Okay, this is done. Yeah. See, as told in the preceding sections, that no, not bit 2, I guess. 99.9% of the base sequence among humans is same. Assuming human genome as 3 into 10 to the power base pair in many base sequence would be the difference. It is these differences in the sequence of DNA which make every individual unique. Understood? That repetition makes everyone different. The change in the single nucleotide polymorphism makes every individual different. But otherwise, if you see, 99.9% of the gene is going to be same for all of them. Okay, same for all of them. Who's this guy dancing in my, class? Okay, it's an advertisement. Okay, yes. Okay. The phenotypic, if one aims to find out genetic differences between two individuals or among individuals of a population, sequencing the DNA every time would be daunting and expensive task. Imagine trying to compare two sets of 3 into 10 to the power 6 base pairs. DNA fingerprinting is very quick way to compare the DNA sequence. So students, DNA fingerprinting is to identify, that okay, this person is the one who is missing. So we are going to start with DNA fingerprinting. Okay, so this person is good, let's say someone is missing or some crime scene happened. We have to make sure that okay, this change in the chromosome is the reason why we are able to identify. Otherwise, imagine every time you go and search your 3 into 10 to the power 6 base pairs, oh, this is the one, this is the one. You cannot do that. So DNA fingerprinting is going to help. Okay, so DNA fingerprinting involves, listen carefully, identifying differences in some specific regions in the DNA sequence called as repetitive DNA. So this repetitive DNA is going to vary from person to person. I told you two person will not have the same set of repetitions. So that is what we are going to identify using DNA fingerprinting. That is, if we understood that this person has, some change, obviously maternal and paternal sequence are going to be different. This person, okay, if I don't draw it in two colors, you people will not even understand. So here, this is one more person. This person also is going to have a sequence that is only special to him. Okay, maternal, paternal. Now, among siblings also, maternal, paternal difference is going to be there. Now, not all the characters from the mother and father will go to the same siblings and it will not repeat after each other. So, look here, because in these sequences, a small stretch of DNA is repeated many times. That's what I told you. Now, let's say AGGAT is repeated many times, like ACGG. AGGAT. It can repeat continuously again, AGGAT, or it can repeat differently. That means I'll show you down, students. Here no place to write. So these repetitive DNAs are separated from bulk genomic DNA. I told you, bulk DNA is the one that we have for 99.91%. This is called as bulk. Okay, bulk genomic DNA. As a different peaks during density gradient centrifugation. That means we can separate the different densities of DNA with centrifugation. Okay, the bulk DNA will be the major peak. That means the highest, how much supernatant we'll have, that the whole thing will be the bulk DNA, and then pillet will be the one who is going to be the repetitive DNAs. The bulk DNA forms a major peak, and the smaller other peaks are referred to as satellite DNAs. Okay, satellite DNAs. Depending on the base composition, AT rich and GC rich, length of the segment, number of the repetitive units, the satellite DNA is classified into many categories like microsatellite, mini satellite area. Microsatellite is usually the area where there is a GC rich area, and mini satellite is an area where there is an AT rich area. Okay, students, are you understanding, or is it too difficult? Tell me, all of you. Any doubts? If you have doubts, ask me right now. Don't keep it later. Okay, this is actually ulta. Okay, that's what I was just checking my notes. Mini is GC and micro is AT. Right, okay, no problem. Chalo. It's not working, students. Once again. I want to show you people an image, but I don't know if I'll be able to show, because all the images are complicated, so complicated that I don't think even you will understand. Actually, don't need that much of information also, but still, like, at least little information. I think this is okay. No, this is also difficult. Okay, never mind. I'll try to make you understand. Okay, I'll try to make you guys understand. So see, depending on this, there are different types of satellite regions. Okay, these sequences normally do not code for any protein, but they form a large portion of the human DNA. Okay, human DNA. Now I'll show you where it is present in the chromosome, but they form a large portion of human genome. These sequences show high degree of polymerization and form the basis of DNA fingerprinting. So before I go to DNA fingerprinting, let's just understand what they just told. Okay. Yes. So students, look here, all of you. Okay, glasses are ready. Okay, now this is a chromosome. Okay, this is a chromosome. This is a chromosome. On this chromosome there are different regions. Okay, on the chromosome there are different regions. Now let me first highlight this. Okay, this is one region. Now in this region itself, okay, in this region itself you will be able to see so many parts. Okay, you will be able to see so many parts. I'll tell you what are those parts. Now these are all some segments. Okay, so many will be there, but right now don't worry about it. Now students, look here, this portion, okay, this portion of the DNA, same color, okay, this portion of the DNA, yes, will have two portions, satellite regions I'm talking about. Okay, will have alpha satellite, okay, beta satellite. Satellite regions depends on the number of DNA strands, tandem repeats and all of that. Now down portion, when you're coming, this portion, it's going to, okay, this is going to form the, okay, microsatellite, okay, microsatellite region. And down, have you always noticed, even when I think Baswa sir taught you, he would have highlighted this like this. Okay, like this. If the color is visible here, like this. Okay, color is visible only, no? This portion is the mini satellite. So they are talking about micro and mini satellite. Okay, this is what you have to focus on. Now students, this understood. On the chromosome itself there will be different regions. Okay, the region in the middle is going to be alpha satellite region, then beta satellite region, then there is going to be micro satellite, then it's going to be mini satellite. All of you understood? Now when we do centrifugation and all of that, we'll be able to get different pieces of it. Now they believe that micro satellite, not believe, they found that micro satellite is AT rich region. I told you this in the starting Chargaff rule when I taught you. I told you both of them do not have to be same ratio. This is going to be GC rich region. Okay, rich region. So here this is going to be rich, this is going to be rich. Now this regions, okay, these regions are going to be short. The microsatellite regions are going to have short tandem repeats. Short tandem repeats. These people are going to have VNTR, that is variable tandem repeats. Variable number of tandem repeats. These two things you try to remember. Okay, now here one more thing we usually teach is size of the repeating unit. Okay, size of repeating unit. What is going to be 2 to 6. Okay, 2 to 6 is the usual range of size of the repeating units. Okay, size of the repeating units, but here it is different. Here it is going to be how much students? It's going to be 10 to 100. 10 to 100 base pairs. Okay, 10 to 100 base pairs. Clear all of you? Yes, let me see the chats. Chalo, understanding ma'am. Okay, no doubts. Perfect. So these are the different regions. Okay, so they are telling depending on the number, depending on how many repeating is there, depending on that, the different types of satellites are there. So you only have to focus on micro and mini satellite. Okay, micro and mini satellite, I've given you an idea. Now see, as polymerization in DNA sequence is basis is basis of genetic mapping of human genome as well as DNA fingerprinting. It is essential that we understand what DNA polymerism means in simple terms. Polymorphism is variation at genetic level arises due to mutation. Recall different types of mutation. I told you frame shift mutation is there, deletion mutation is there. New mutation may arise in individuals either in somatic cell or in germ cells. That is in the normal our vegetative cells, no, like we tell vegetative cells like that in our somatic cells, or germ cells, which is sexually active reproducing cells. If germ cells mutation does not seriously impair the individual's ability to have offsprings who can transmit mutation, it can spread. Okay, it can spread to the other members of the population through sexual reproduction. Now, all this DNA polymorphism, if more than one variant at a locus occurs in a human population with a frequency of 0.01, in terms it's an inheritable mutation. Okay, it's observed in a population at higher frequency, so it is referred. So students, this is a little complicated. Usually we don't stress a lot about it, but we stress a lot about this. Okay, because this is what questions usually come. All that inheritable mutation, all we rarely see it. Okay, so here students, see, the technique of DNA fingerprinting was initially developed by Alec Jeffrey. He used a satellite DNA as a probe that shows a very high degree of polymorphism. Now, it was called as variable number of tandem repeats that is present in your mini satellite. Yes or no? Mini satellite. So the technique was earlier, involved in southern blotting hybridization. So what? All this you forget. How we will do DNA fingerprinting. Okay, imagine one crime has happened and we got the semen or the saliva or something we got. Okay, semen, saliva or something we got, students. When we got what happened to the video? Clear only, no? Yeah, clear. Okay, when say we got something that will identify the DNA, we will do isolation of DNA. Yes or no? We'll do isolation of DNA. Then what we will do, we will digest the unnecessary sequence, the 99.9%. Do I want it to identify who is the culprit? I do not want it. So I will use restriction endonuclease and cut the inner corners. Okay, I'll cut the inner corners. Okay, using restriction endonuclease, I'll cut the inner corners which I don't require. Then separation of DNA fragments by electrophoresis, which I'm going to teach you today. Okay, which I'm going to teach you today. Electrophoresis, separation of DNA fragments by electrophoresis, where this will be your agarose gel. Okay, this will be agarose gel. I don't know, students, has Baswasa taught you in biomolecules? I mean, we don't teach like that in agarose gel, but has he taught you anywhere agarose gel? So students, this is your DNA molecule. So we'll see that few people are running faster. These are the larger ones will not run too fast. The smaller ones will keep running faster. Okay, the smaller fragments will run. So this is totally done on the base of size. So we'll do electrophoresis. Then we'll transfer that electrophoresis blotting of separated DNA fragments to a synthetic membrane called as a nitrocellulose membrane or a nylon membrane. Okay, we will do southern blotting here, where we will transfer those separated DNA fragments into a nitrocellulose or a nylon paper, like a nylon sheet. Now hybridization means what? Okay, I understood that this is the sequence I wanted. I will use a VNTR probe. VNTR probe is nothing but a complementary. Now imagine, students, let's go down. Okay, this is one thing. So imagine I got a sequence. Okay, the sequence was AGAT. Okay, this is the sequence that is making a difference for me to identify who is the culprit. Now what I'm going to do, after I got it in the electrophoresis, after staining it, after seeing it, I will use a VNTR probe. Okay, VNTR probe, which is a variable tandem repeat. This is going to be complementary to this. That means it's going to be TCTA. Okay, this is going to be complementary to this, and this is going to be radioactively labeled. Radioactively labeled. So since this is radioactively labeled, what will it go? It will go and bind with this fellow, and it is going to be shown to us. Yes or no? Radioactive labeling, why do we do? So that it will be seen to us. So like this VNTR probe will go and attach there. This way we'll be able to confirm the results that we got. Okay, confirm the results that we got. So look here, detection of hybridized DNA fragments by autoradiography, because we radioactively labeled it. Now we'll be checking it under autoradiography, and then we will be able to get the DNA fragments. Now there is a crime given here. Okay, see, I try to understand from the image. Okay, students, this is individual A, this is individual B. Two individuals are there, and we got a, so the crime happened where we got a blood. Okay, so much blood from the criminal was C. Okay, I mean we got a blood sample of someone who is going to be C. Okay, now during the murder, the person died. Okay, that person's blood we got. Now this is what DNA we got. This is the DNA material we got from the crime scene. Okay, from crime scene. Now I have to find out the C that I got. Okay, is it from the individual B or is it from the individual A? That is my task. Okay, because I am trying to find the evidence. I am the CBI, so I have to find evidence. Yes or no? I have to find evidence. So if I have to find evidence, I need to make sure that is this DNA material, whose DNA material it is. To find whose DNA material it is, what I'm going to do, I'm going to check. See students, paternal chromosome, maternal chromosome. Maternal chromosome obviously is going to be different for all of them. Okay, here you can see 3. I'm just going to count. Okay, repetitions 4. Okay, this is going to be 1 2 3 4 5 6 7 8 9 10 11. My god, 11 8 4 no 9 9. Okay, here 9 8 1 2 3 4 no 7. Okay, here also 9 and 7. Okay, the bulk, some bulk DNAs also will be there. See, 1 2 3 4 5. Okay, 11. This is going to be okay, I am CBA. Okay, 1 2 3 5 and 2. Thanks for accepting, students. So this is the repeating, we got the numbers. Okay, now obviously paternal and maternal is going to be so different. We can see here, here it is 11 and 9, here it is 3 and 4 from the individual. Now let's see what is the blood sample that we got from the crime scene. Okay, crime scene, students, we got a blood sample that had, so this is electrophoresis, gel electrophoresis. So you can see the smaller fragments have ran before, the larger fragments are in the backside only. Okay, now when I'm trying to align, okay, when I'm trying to align, I notice that this is 11. Okay, this is 11. Yes, so I was just thinking if it was loud. Okay, okay, so students, here it was 11. Okay, 11. Here it is 9. This is going to be chromosome number 7. This is going to be 8. Okay, this is going to be 1 2 3 4 5 6 7 7. Okay, this is 5 2. Okay, this is the chromosome. So this is the crime scene. Say DNA we got was this one. Now you have to see which is going to align. Students, who is the criminal? Looking at it, you will tell. But I'm telling, how will you identify? You have to identify. No, so this is going to stop at 11. Can you see this is stopped at 9? So like this numbers is going to be there, and they're going to run till there, and they're going to stop there. Okay, they're going to run till there, and they're going to stop there. Tell me, who is the criminal? Who is the criminal? CBI season 1. This is the 8, no? A or B? Who is the criminal? Can you count, students? I am. Who is the criminal? Okay, the criminal is B. Students, can you identify like this if I draw? Okay, 7. Okay, See, I'm going to draw one. You have to wait until I finish drawing. After that, you identify and tell me who is the criminal. Okay, this is maternal, this is paternal. Okay, then we go for chromosome 7. Okay. Wherever I put dots, you have to count. Okay. So, students, actually, this is the task. I cannot do this much. Okay, it will take a lot of time. So, just tell me in the gel electrophoresis, at what position is Roy's? So, let's say this is the gel electrophoresis. Okay. 1, 2, 3, 4, 5, 6, 7, 8. 1 second, I have to draw a little more bigger. 9, 10, 11. Okay, so tell me where will Dawn's, the first maternal fragment is going to be there. Answer quickly. Where is Dawn's maternal fragment going to be there? People are so lazy to answer. Quickly. Tell me where is Dawn's going to be there. If you want to help, I will count and tell you. If you want help, I'll count. If you don't want help, see 1, 2, 3, 4, 5, 6, 7. 7. Okay. 1, 2, 3, 4, 5. 5. Tell me. People are so lazy to count. I've only counted. Tell the answer. Yes, answer. Chalo. Answer. That is C, 7. Okay, Dawn's is going to be somewhere here. Dawn's DNA fragment is going to be somewhere here, maternal, and paternal is going to be somewhere here. Okay, so this is how you will identify. Now Roy's is 4. Okay, 4. So where is it going to be? It's going to be here. Okay, here. Understood? Like this, you will be able to identify this. This is gel electrophoresis, and here wells are going to be there. Okay, where we'll add the things. Okay, where we'll add the things. So this is how it is going to happen. Understood? So wells are going to be there. Full closing will be there. They'll be kept in a like a tumbler where there'll be liquid filled, and this will be, so what happens, the movement happens. Okay, some does not move, students. Now let's say Roy's was like this only, means same it was, means we'll tell it is undigested. Understood, all of you? So this is your DNA fingerprinting. We have completed DNA fingerprinting. So this you have to remember. Okay, this once again I think I have to teach you. So here, first thing is you do isolation of DNA. That is, let's say this is the strand of DNA and you want to take only this sequence. Okay, And what do? Second thing, okay, first you isolate the DNA fragment. The isolation could have done from, it could be from semen, it could be from nails, it could be from hair, it could be from bones, okay, bones, etc., etc. You have isolated the DNA. Then you are going to do chopping. Okay, chopping you're going to do. We're going to do cutting. Okay, so that is going to be with endonuclease. Which endonuclease? Restriction endonuclease. Okay, restriction endonuclease. Clear? Once again. Right. One minute, dear students. You just give me 1 minute. One second. Yeah. So many office calls are coming. That's why I just told them that I'm in class. Okay, students, restriction endonuclease. We will cut only a small portion of the DNA which I need. Okay, my desired, right? Desired. After that, after getting my desired, what am I going to do? I am going to do gel electrophoresis. Okay, gel electrophoresis. After doing gel electrophoresis, I'm going to run, okay, with the help of agarose gel. Okay, agarose gel. I'm going to run the gel electrophoresis. After that, I'm going to do southern blotting. They have not told which blotting. It can be southern, western blotting. Okay, different types of blottings are there. So we will do just blotting. Okay, and just get the copy of it on a nylon or a nitrocellulose sheet. Okay, after the nylon nitrocellulose, we will do use a VNTR probe. VNTR probe, as I told you, it is going to be complementary to the strand we got. Okay, it's complementary to the strand. Then that we are going to check under audio imaging, auto radiography. Done. These are the steps. Okay, the steps, if you forget, student, then gone. Okay, gone for a toss. Now anything else, all of you? Yes, blotting, VNTR probe label. Very nice. Okay, very good. Done. That's it is your DNA fingerprinting. Okay, that's it is your DNA fingerprinting, and that's it to do with all of your this one also. What is that? Your molecular basis of inheritance. Any doubts you can ask me right now. Okay, I will wait, or I'll ask you guys some questions. Transcription, I taught you guys everything, no? Yeah. This translation also done. Any doubts? Tomorrow, 12 o'clock. Biotechnology at 12 o'clock tomorrow. Okay. Biotechnology tomorrow at 12 o'clock. What I'll teach you in biotechnology, I'll teach you about this better. Okay, I'll teach you about gel electrophoresis. I'll teach you about how separation happens. I'll also teach you about, different types of plasmid. And then on Saturday, we will finish the application is what I'm believing. We'll try to finish the application chapter as well. Okay, it will be one shot. So we will try to finish it in 3.5 hours, like in detail. So tomorrow's class is going to be little more like a little more complex topics, but we will definitely try to learn a lot easier. Okay, a lot easier. Now students, if you look here, okay, this is this one, okay, a molecular basis chapter. So here we have done, so we have done all this. Okay, we have done this regulation, we have done. Okay, from starting, if we go, this is a big chapter. So excuse me. Okay, so molecular basis of inheritance. See, we understood what is DNA. Okay, structure of a polynucleotide chain, very important students. Don't even think of skipping this. Here you should know why the 5-phosphate came, here the hydroxyl came. If you want detailed lecture, you can check on Vedantu NEET English. I have done it. Okay, I've drawn the diagram and taught. Here I cannot do that much because of the pen. So here backbone, I told you, adenosine, then the salient features of DNA, then the bonding, how they bind and how many hydrogen bonds are formed between each other. Okay, then so these two will project outside. Okay, these two are the strands. These adenine and thymine will project outside, and that also I told you, right, how they will be there. And this is the central dogma. On this basis only the entire chapter is going around. Okay, then packaging of DNA helix, such a big strand, how do we pack it inside? So here you can see how it looks under the microscope. I could not show you this yesterday. Look here how it looks under the microscope. Can you see dot? Those dots are nucleosomes. DNA plus histone octamer forms the nucleosome, and they are kept together with the help of a linker protein, which is called the H1 protein. Okay, H1 protein. And then we studied about chromatin. We studied about euchromatin, heterochromatin. Depending on their staining, they are U and hetero. U is light, hetero is dark. Okay, this is like a Gopika ma'am express. Okay, and then search of genetic material. We started with Griffith's experiment, where we worked on Streptococcus pneumoniae, and he injected R strain and S strain to the mice. S strain was virulent, so obviously the mice died. Then he heat killed the S strain, and he noticed that something did not get completely damaged. When he did heat kill S strain alone, the mice lived. But in later, when he mixed S strain and the R strain, he noticed that something got transformed from the S strain to R strain, and that he called it as a transforming principle. Okay, then to check the biochemical nature of the transforming principle, Avery, MacLeod and McCarthy came into the existence, and they checked the biochemical nature, where they used RNAs, protease and DNAs, and they tried to find out what went wrong. But it was not widely accepted. Then the genetic material is DNA was proven by your Hershey and Chase experiment, where they used a bacteriophage, that they used E. coli and they infected it. Then they got to know that the pillet made the DNA was there and supernatant was not there. All of that. Properties of genetic material, the criteria to become a genetic material. You cannot chumma become a genetic material. You should go through a criteria to become the genetic material. Okay. Is it cropped? No. Now then we understood why the DNA is chosen over RNA. Then RNA world, where we understood that RNA is not waste. It is going to do the job of catalyst. It's going to act as a tRNA. It's going to act as a messenger RNA. It's going to act like a catalyst. All this we learned. Then students, this is very important about the semi-conservative nature of DNA, where we understood that the parent DNA and the daughter DNA, the parent will act as a template to form the daughter DNA. Then the parent and daughter will become together, and that will form a hybrid. So you have to know the number. Yesterday I made you do the number. In the first generation is going to be 100% hybrid, second generation is going to be 50-50, third generation it is going to be 75-25, 25 will be hybrid, and the fourth generation will be 12.75 to 87, like that calculation. Okay, like that. I told you easy method to solve that as well. And what else, students? Machinery. Okay, machinery, that is how is this DNA replication happening, which are the which are the enzymes that is helping in unwinding, opening and zipping, cutting, chopping. All this we studied. Okay, all this we studied as well. Then, so 4 enzymes you have to remember, okay? Helicase, primase, ligase, and DNA polymerase. DNA polymerase is the most important because that is mentioned in your NCERT, and even ligase is mentioned in your NCERT. Helicase is actually unwinding enzyme. Like this, the complementary strand, this fellow will help to unwind. Then unzip also we can do. Unzip, then the DNA polymerase will form the new strand. DNA ligase will join the Okazaki fragments and all the fragments. Primase will lead the DNA polymerase. Okay, then transcription, students. Transcription very easy. You have to remember the units of transcription: promoter, structure, gene, terminator. The processes are going to be initiation, elongation, and termination. Okay. Then DNA dependent RNA polymerase is there. Template strand, coding strand. Coding strand is same as the RNA, just that thymine is replaced by uracil. Okay, then upstream, downstream. All this I taught you yesterday. Polycistronic, monocystronic. Monocystronic is seen in eukaryotes, polycistronic in prokaryotes, introns, exons. Introns are present which are non-coding, exons are there which are coding. All this we studied. Splicing, we studied capping, we studied tailing, we studied. This is your entire transcription where RNA polymerase will combine with sigma factor. It will go around and it will go reach the structural gene. Structural gene is a small region of the gene which is going to become mRNA. It will form the mRNA. And then to stop this RNA polymerase, rho factor will come and bind, and this rho factor is going to rho factor is going to bind with this RNA RNA polymerase and terminate the reaction. Okay, terminate the reaction. Then student, 3 types RMT I told you, 1, 2 and 3. Tailing, capping I told you. Then genetic code 4 into 3, 4 to the power 3, that was 20, what is that, 3 triplets are going to code for 64 codons. Okay, out of that 61 is going to code for amino acid, 3 are going to be nonsense codon or stop codons which are not going to code for any amino acids. Okay, and then they are universal degenerative, they are read in a continuous fashion, and their initiator codon is there. Then sickle cell anemia we spoke about. Okay, we spoke about insertional and deletion, tRNA. Okay, I think this I missed. Okay, tRNA has an anticodon loop. I told this, but I'm just telling I missed the paragraph. Okay, and then students, remember, for us there is going to be a tRNA who's going to start the reaction, and that tRNA is going to be called as initiator tRNA. Okay, and then translation, 2 steps are there: charging and amylation. I showed you this. Untranslated region, release factor, gene regulation where the gene can decide to switch off. Then lack of prone concept, this I did not put because I explained this. So basically same thing, only if lactose is there, then the repressor protein is going to not go and bind with the operator. If lactose is absent, repressor will go and bind with the operator and stop the regulatory proteins, and they are not going to secrete anything. Okay. Yes, negative, students, I forgot to tell you this. If lactose is present, it is going to be positive regulation. If lactose is absent, it's going to be negative regulation. Okay, negative regulation. So both will happen in lacopron. Okay, both will happen in lacopron. Then HGP project, DNA fingerprinting, salient features, and repetitive satellites and satellite DNAs, polymorphism. Polymorphism, actually we don't give that much, we do not tell a lot, but yeah. PCR, students, here amplification we are going to do with the help of PCR. Okay, this you'll study tomorrow, that's why I'm not focusing in today. Yes, that's the end of your molecular basis of inheritance. Okay, summary is here. So done. Okay, done and dusted. The chapter is done. I hope all of you enjoyed the session. See you all in the next class. Thank you so much for joining. Do not forget to like and comment and subscribe to the channel without fail. And thank you so much. See you all tomorrow with biotechnology part 2 in Vedantu NEET English channel. See you all.